In this video, let's discuss about bacterial flagella. With the help of flagella, the bacteria can move from one place to another, and therefore they are called as organ of locomotion. The length of the bacterial flagella ranges from 5 to 20 micrometers, whereas the thickness of the flagella ranges from 0.01 to 0.02 micrometers. The bacterial flagella are attached to the bacterial cell wall and are arranged in various ways. Those different ways of flagella arrangement are monotrichous, lophotrichous, amphitrichous, and peritrichous. The bacteria with monotrichous flagellar arrangement have a single flagella at one end, whereas the bacteria with lophotrichous flagellar arrangement have a tuft of flagella at one end. The amphitrichous flagellar arrangement consists of two flagella, one at each end of the opposite poles, whereas the peritrichous flagellar arrangement consists of flagella present throughout their body. The bacteria with monotrichous flagellar arrangement are Vibrio cholerae and Pseudomonas. The lophotrichous flagellar arrangement is present in Spirillum. The amphitrichous flagellar arrangement is present in Alcaligenes faecalis, whereas the peritrichous flagellar arrangement is present in Salmonella typhi and E. coli. Now let's have a look at the various structures present within the flagella. This may look complex, but it is very easy. It has the following parts called the filament, hook, L-ring, P-ring, outer membrane, the rod, the plasma membrane, S-ring, M-ring, and the whole part which is embedded within the cell wall is called the basal body. These ultrastructures of flagella are appreciable under electron microscope. While summing up all these individual structures, we get three major parts of flagella, such as filament, basal body, and hook. The filament of the flagella is the longest portion and consists of a hollow cylinder which is made up of a protein called flagellin. And the basal body of this flagella is the one which is embedded inside the cell wall. The basal body has 2 to 4 rings which are connected to the central rod. Usually, gram-negative bacteria have 4 basal body rings, whereas gram-positive bacteria have 2 basal body rings. Hook is a flexible structure which is responsible for connecting the filament to the basal body. We have two different ways to detect or demonstrate the flagella. Those two different ways are direct and indirect. The direct ways through which we demonstrate flagella are tannic acid staining with the help of Leibson's method or reuse method. The second method of direct demonstration of flagella is by phase contrast microscopy or dark ground microscopy. The indirect means by which we demonstrate the motility or the flagella is by Craigie tube method, hanging drop method, and also with the help of a semi-solid medium, for example, mannitol motility medium. And that's all the detail about the flagella. I hope you guys found this video useful. Do like this video and subscribe to Medinare for more. Have a nice day.